So it's a great pleasure to welcome our first speaker, Dr. William Evans. Dr. Evans is a full member and full professor at St. Jude Children's Research Hospital in the Department of Pharmaceutical Sciences, University of Tennessee College of Medicine, and he's going to tell us about his work in leukemia.
Thank you, Madam Chairman. Okay, if we can get the first slide up. I'm going to talk about work that's ongoing in the Center for Precision Medicine in Leukemia. If I can get the first slide to go here. You got that? And I understand there's going to be a conflict of interest slide, comes up first and stays on for as long as it wants to. Yep. And I can't do anything about this. So the Center for Precision Medicine in Leukemia is really an extension of work that's been going on at St. Jude for a decade or more. And it was a formal way to integrate work across several departments and across several institutions in the U.S. to bring it together. And the big change for us was to add studies of adults in addition to children with acute lymphoblastic leukemia. So the project includes a genome-wide look at somatic and inherited variants in Project 1 that are related to event-free survival or other measures of response, general measures of response. Project 2, which I'll talk about today, is getting a bit more granular and trying to look at resistance, de novo resistance or acquired resistance to individual anti-leukemic agents that will be used to treat those patients. And the third project, which Mary leads, focuses on trying to understand inherited genome variants influencing drug toxicity. And one of the strengths of the center is that there's enormous overlap across these three projects in patients being studied. So we only have to do whole exome sequencing or genome-wide SNP analysis, et cetera, one time on that cohort of patients that's useful to all three of the projects. And it involves a number of investigators across the country. So Project 2 is really focusing on the left side of this slide, which is trying to identify both inherited and somatic genome variants that influence sensitivity to drugs and therefore might influence cures of ALL. Oh, she wants me to use the mouse. Okay. And then Project 3, as I said, looks at those same variants, inherited variants that influence susceptibility to toxicity to antileukemic agents. So today, in my 25 minutes, I'm going to use two drugs, glucocorticoids and thiopurines, Macaco Purine, to illustrate sort of how we're going about this and what we've learned. So we're using a strategy that's familiar to everyone in this room. Most of us are using similar approaches where we're, in our case, interested in identifying both germline and somatic or leukemia cell genome variants that might influence our phenotype of interest. In Project 2, it's the LC50, or the sensitivity of leukemia cells to individual antileukemic agents. We thought going in, and it's proven true, that it could be either inherited or somatic genome variants that would influence the de novo or acquired sensitivity to chemotherapy, whereas toxicity is driven more by inherited genome variants that influence other tissues. And so we're interrogating both of these genomes in all the patients being studied, and we go from association and replication to then pursuing mechanism to get a bit more confidence in the finding being real, and then ultimately are interested in translating that to the clinic as diagnostics. So we've been working on either side of this genome variation in leukemia for many years, both germline and somatic. And so what I'm going to do is use a couple of recent papers to illustrate the somatic genome variants that influence both glucocorticoids and Macaco Purine. And then at the end, I'll come back very briefly and connect this to an inherited genome variant that also influences a patient's sensitivity to thiopurine medications. So again, our strategy is this. We take leukemia cells from a bone marrow diagnosis. We grow them in a 96-well plate. We expose them to different concentrations of anti-leukemic agents, including some of the newer targeted agents. We then interrogate the leukemia cells and the germline genome using several strategies from whole exome or whole genome sequencing in some cases, but also looking at methylation, gene expression, microRNAs, et cetera, attempting to identify the subset that are influencing the sensitivity to individual agents. And of course, we're always interested in knowing whether that is also related to disease outcome, event-free survival. So AIM 1 and 2 are really focused on two different types of drug sensitivity. One is de novo, that is before they've seen any treatment, and the other would be acquired, that is perhaps there are either selections of subclones that existed at diagnosis that carry variants that confer resistance to some of the agents, and that's what causes patients to relapse from their treatment, and so we're interested in that acquired resistance as well. And I'll give you a couple of examples in the next few minutes. As I said, we're interested in mechanism. And the new thing here is that we're studying an equal number of adults with acute lymphoblastic leukemia as well as children. The reason we're pursuing the adult question is that it was recently shown by our collaborator, Wendy Stock, a few years ago, that if you take adults with ALL and you have them treated on childhood ALL protocols by pediatric oncologists, that their outcome is better. They're going to get more intense therapy, and they're going to get more of that therapy, and their event-free survival is improved, as shown here. And that's been replicated in a couple of groups in Europe and UK as well. However, if you compare their outcome, even treated by pediatric oncologists with pediatric trials, children still do better than adults. And so one of the questions we're trying to answer is, why is that true? Is there a difference in de novo or acquired resistance to chemotherapy that causes some of that, and what can we do about it? So this is an example of glucocorticoids, a work that we published some time ago that we're trying to build on, and it makes the point that if you do this study at diagnosis, you see about over 1,000-fold range of sensitivity to prednisolone or dexamethasone at diagnosis. So enormous differences among patients with the same disease, and it has been shown by our collaborator Rob Peters, but by us and others since, that this de novo sensitivity is also related to disease outcome, event-free survival, as shown here. So we have just gotten underway in this project, starting to study adults as well as kids. These two histograms show sensitivity to prednisolone at diagnosis, kids on the left, adults on the right. I point out there's only 25 adults. We're now accruing patients rapidly to this, but you can see already there's a suggestion that a higher percentage of adult ALL cells at diagnosis exhibit relative resistance to glucocorticoids, 65% in this small subset versus 25% of adults in a much larger cohort. So we initially used gene expression in leukemia cells to try and lead us to genes that might be altering or influencing sensitivity to steroids. This is a work published over 10 years ago now where we were able to identify a relatively small number of 35 or so genes that were influencing sensitivity to glucocorticoids. This allowed us to focus in on some of these, like overexpression of an anti-apoptotic gene, MCL1, which was subsequently replicated by the Boston group, or downregulation of a chromatin remodeling complex, which you can certainly mechanistically connect to glucocorticoids mechanisms of activity. And so what we have been doing more recently is trying to expand the cohort in kids and adults so that we can take a deeper look to perhaps identify additional mechanisms, genomically driven, of resistance to glucocorticoids. And that led to a recent one that was published last year, which identified a couple of genes that were being differentially expressed that altered de novo sensitivity to glucocorticoids. Those two genes, the only two genes in that subset of genes that were in the same pathway, was the gene encoding caspase-1 at top and NLRP3, which is the protein that activates caspase-1 from procaspase to caspase. The mechanism turned out to be an epigenetic mechanism. Those leukemia cells overexpressing these genes had hypomethylation of the promoters of these two genes, and therefore allowing them to be expressed at a higher level. And the question became, what is activating caspase or overexpressing caspase-1? What is the target protein that would lead to steroid resistance? A postdoc in my lab, Steve Powell, took on this project. The first thing he did is an in silico look and searching the literature and identified this paper a number of years ago that said the androgen receptor was cleaved by caspase-1. And looking at the motif where that cleavage occurs, LLDD, in silico review of the amino acid sequence of the glucocorticoid receptor revealed a very similar motif, LLID. And so the experiment was to take recombinant caspase-1 incubate it with the glucocorticoid receptor and ask whether it cleaved the glucocorticoid receptor, and it did. It does so in the transactivation domain, so it's when that cleavage occurs, it's killing that receptor. We showed that we could block that with either a tetrapeptide inhibitor of caspase-1 or a small molecule inhibitor with the recombinant protein. That then led to the experiments that said, okay, if we take human leukemia cell lines, overexpress caspase-1, activate it, does that confer resistance to glucocorticoids? You can see on the far right at the bottom that there was some 7 to 15-fold increase in the amount of glucocorticoid, either DEX or PRAD, required to kill these leukemia cells overexpressing caspase-1. And it was also associated with a concomitant decrease in the amount of cellular glucocorticoid receptor. That led us to do some experiments to see if we could, in fact, see some pharmacologic or biologic effects of this cleavage of the glucocorticoid receptor. We did ChIP-seq as well as looking at steroid induction of gene expression. And with the ChIP-seq experiments, we were able to show a threefold reduction in the number of reads that were coming down in the caspase-1 overexpressing cells, entirely consistent with a reduction in cellular glucocorticoid receptor levels. Good thing was what was coming down was still binding to the appropriate motif for the glucocorticoid response element by steroids. That then led to some experiments where we said, okay, if we take these caspase-1 overexpressing cells and expose them to low doses of steroids, it should induce gene expression for those with a GRE. We did that. We identified the 50 most upregulated genes after exposure to glucocorticoids, and then we compared that in blue to the cells that were overexpressing exogenous caspase-1. And you can see in 48 out of those 50 genes, there was blunted transcriptional upregulation in the caspase-1 overexpressing cells. Again, consistent with there just being fewer receptors in those cells overexpressing caspase-1, and we could see this at the protein level as well as at the mRNA level. So to summarize sort of this finding and what we're trying to do next with it is that leukemia cells overexpressing these two genes lead to cleavage of the glucocorticoid receptor, making them relatively resistant to glucocorticoids who require the receptor for their pharmacologic effect. It blunts transcriptional activation. It increases resistance to steroids, shown here, and I've shown you that data. And I haven't shown you the data, but this then relates to their response overall to treatment. It sort of begs the question, what can we do about this? I mean, we're interested in finding mechanism, but we're interested in doing that for reasons. One of which is, it begs the question, could we use a caspase-1 inhibitor in these patients who have resistance on the basis of its overexpression and reverse that resistance to glucocorticoid? We did sort of the proof of principle experiment, which was to overexpress the cowpox virus protein CRIM-A, which is a known caspase-1 inhibitor. When we did that, we increased the amount of cellular glucocorticoid receptors in those caspase-1 overexpressing leukemia cells. And as you can see, we had a big effect on the LC50 for glucocorticoids, increasing sensitivity about 40-fold. So to me, this was an initial proof of principle that this would be a way to reverse resistance in these cells and in these patients. We're now pursuing small molecules that could do this. We can't express that cowpox protein in patients. There are caspase-1 inhibitors in clinical trials by Vertex, but those clinical trials were subsequently stopped. When we tried to do this in cell systems in the lab, we couldn't get enough of this drug inside cells to inhibit caspase-1 and reverse resistance, and that may well be the problem with those drugs. So we're now collaborating with our colleagues in chemical biology and therapeutics, Kip Guy and others, to look at other small molecules or derivatives of existing small molecule inhibitors of caspase-1 as a strategy. So how common is this mechanism of steroid resistance? Well, in our patients and kids, it was about 23%. So it doesn't explain all glucocorticoid resistance in leukemia. I didn't anticipate that would be true, but it's a pretty big fraction of the subset of patients who are resistant to glucocorticoids. We don't know if this mechanism is operative more commonly in adult ALL. That's one of the questions we're pursuing in the center. I would also point out there are many ways that cells can become resistant to glucocorticoids. Some of those have been recognized for years. and we're basically adding to that to try and understand across the whole spectrum of childhood or adult ALL what's driving resistance at the genetic level, somatic and inherited. I will say that this caspase-1 NLRP3 mechanism connects back to one of the first mechanisms published from a clinical analysis of patient leukemia cells, where our collaborator Ching-Han Pui, as a fellow, published in 1982 that newly diagnosed cases of pediatric ALL that had low levels of glucocorticoid receptors had worse outcome. And this is a mechanism by which that appears to happen in a subset of patients. So we think there are other mechanisms. We think there are also other genomic sources of genome variation that influence sensitivity to prednisolone as well as other agents. So as I said earlier, we're interrogating other genome variants or microRNA expression differences, copy number alterations, CpG methylation across the genome, attempting to identify in a broad way all of those genome variants influencing steroid sensitivity in patients. And we're working with our statistical group in the center to see from that group of 500 variants that are related to de novo sensitivity, can we identify the subset that explains the greatest variation in steroid sensitivity, and we can reduce that down to 39 features to create the most parsimonious model that, even after correcting for bias, explains around 40% of the variation in LC50. You can see it's a mix of mRNAs in leukemia cells, copy number variants, CpG methylation, microRNAs, and SNPs in this model. So we're obviously interested in doing this for other drugs besides steroids, and we have postdocs and scientists in the lab working on other agents as well as targeted agents that are used to cure 90+% of kids with ALL. So acquired resistance, just a couple of slides about that. We were curious if this mechanism of caspase-1 overexpression was operative at relapse, and so we studied diagnosis relapse pairs, that is leukemia cells from patients at diagnosis and then again a few years later when their disease recurred. And we show, first of all, they're more sensitive to steroids at relapse. We also were able to show that upregulation of caspase-1 and NLRP3 is quite common in relapse when compared to diagnosis in these paired cases. Switching drugs just a little bit to thiopurines, we're also wanting to ask the question about acquired resistance to mercaptopurine. And you can see here again diagnosis versus relapse pairs of leukemia cells. There tends to be greater resistance in a recurrent disease than at diagnosis, which may in fact be part of the reason for the relapse. In work that we did in collaboration with Bill Carroll at NYU, looking at somatic genome variants in leukemia cells at relapse, there were several hotspots in the gene NT5C2, which is a nucleotidase that cleaves phosphates off of the triphosphate or monophosphate of thiopurine active metabolites. You can see this hotspot at codon 367 when overexpressed in leukemia cells makes those cells considerably more resistant to thiopurines, thioguanine in this case. And one can also show that this is an activating mutation, that it increases the dephosphorylation of the active metabolites of thiopurines in those leukemia cells that have acquired this genome variant. We don't see this as an inherited variant. It's only a somatically acquired genome variant, but it leads to markedly lower active drug in leukemia cells in patients who have acquired that variant in their leukemia cells. So, to sort of head toward the end of my presentation here, I wanted to connect this a little bit to an inherited source of variation in thiopurine sensitivity. Led by June Yang, a colleague in the center and a co-investigator on a couple of these projects, we did a GWAS of 6-mercaptopurine tolerable dosage at steady state among children with ALL to see if we could identify genome variants that would be responsible for some patients being unable to tolerate the full dosage of mercaptopurine. And there were 2 hits that reached genome-wide significance in this analysis. I was happy to see that TPMT was one of them, but there was a new variant that also appeared as genome-wide significant. I will say that the difference from this study and what we'd done before was that this was a very ethnically diverse population of patients of European, African, Native American, and Asian ancestry. And that's important because this NUD-T15 variant, as I'll show you in a minute, it's pretty uncommon in European and African ancestry individuals. Shown here is individuals who inherit either homozygous for a deficient allele of TPMT or heterozygote, and those also with this new variant of NUD-T15. As I'll show you in a minute, NUD-T15 is also an inactivating enzyme for thiopurines. So this variant, inherited variant, leads to loss of function, and that leads to the inability to tolerate full dosage of 6-mercaptopurine. They require a much lower dosage of the drug, and the heterozygotes, as was seen with TPMT, are intermediate in their tolerable dosage of vincristine. Now, this variant in NUD-T15 had been observed previously, only recently, in a study of inflammatory bowel disease patients in Korea, in an Asian population, where in their genome-wide analysis, it had a p-value smaller than anything I think I've ever seen, 10 to the minus 94. So it was a very highly penetrant, relatively common in Asian population, inherited variant, missense variant in NUD-T15 that alters thiopurine sensitivity. These are the data for allele frequency in these different ancestry subsets of the population, very rare in Africans, very rare in Europeans. I think that's why we never saw it in our previous studies of thiopurine intolerance, whereas it's more common in people of Native American ancestry, Hispanics of Native American ancestry, as well as Asian populations. And so if you look across those populations, Asians, Hispanics, and Europeans, you can see this association between inherited genotype of NUD-T15 and tolerable dosage of 6-MP. I would point out that there's only one heterozygote in the European cohort here that did have lower tolerance, but we didn't see any homozygous variants, and it's all driven by the allele frequency. So you put these two together, TPMT and NUD-T15. If you're heterozygote for one, TPMT, we knew you could tolerate a lower dose. You had to receive a lower dose. That's also true for NUD-T15. If you're het for that, you need a lower dose of 6-mercaptopurine. If you're het for both, then you need even a lower dosage. You've lost two inactivating pathways, at least one allele of each. And then if you're homozygous for either, you have lost even more of the inactivation pathway. We have never seen a patient homozygous for both. I'm sure that patient's out there somewhere. It's just a matter of not having seen it. Now, also led by June Yang, we did some additional sequencing in these patients that had thiopurine intolerance, and were able to identify 3 additional loss of function alleles for NUD-T15 that we were able to show were associated with loss of function, as well as low activity in vivo and poor tolerance to thiopurines. So to put all this together, NUD-T15, TPMT, and the acquired variant in NT5C2, 2 of these, TPMT and NUD-T15, are inactivating pathways, and these are loss of function variants. People that inherit those have much higher intracellular concentrations of active drug and much more cytotoxicity, therefore require a lower dosage. This has been known for TPMT for decades and NUD-T15 more recently. Alternatively, individuals whose leukemia cells acquire an activating mutation in this inactivation pathway now inactivate the drug more rapidly in their tumor cells, not in their normal tissues, and this is a mechanism of acquired resistance to thiopurines. You can't get enough of active drug in their leukemia cells because of the revved up inactivation pathway. So obviously, we're looking, when a patient walks through the door, we're not sure whether we're seeing loss of function inherited variants, acquired variants of inactivation pathways in their leukemia cell, or perhaps a mixture of those. So obviously it's important that we interrogate each of these as we go forward in trying to develop precision medicine strategies, in this case for thiopurines. And so it is complex, but it's logical. And to me, when there's logical complexity, I can make that work. And part of what we're trying to do in the center is identify some of these other genomic drivers of drug sensitivity and integrate that into a more comprehensive strategy. So I'm going to wrap it up there, acknowledge the fact that there are lots of people that have contributed to this in the center and at our colleagues at UCSF and other sites in the Children's Oncology Group, and obviously our patients who agree to participate in all of these clinical trials and studies. So I'll stop there and thank you for your attention and about your questions.
If you have questions, please come to the microphones in the center. I don't know, is my micro? There we go. I'll just start. I always learn something. You flashed by very quickly the slide where you tried to come up with the percent variability in LC50 to glucocorticoids explained by the different types of genomic variation. Because I think for cancer, it's really interesting to think about how much is accounted for by the germline and how much is somatically acquired. So what did the germline SNPs contribute to that? It was
there were a number of SNPs, like 12 or 15, that were part of that. And that was a forward selection model where you add the variant that explains the most variation, then you add the next one and the next one, looking for at least a 1% improvement in the ability to explain variation in phenotype, in this case, steroid LC50. So we haven't published that yet, beware. And we haven't looked at it from the percentage-wise of how much of that was somatic versus inherited. My impression is it's a bit weighted more towards somatic for these phenotypes.
Yeah.
It's not going to be for drug toxicity, but I still believe, the inherited SNPs are contributing probably a third or so of that model.
Kathy.
Yes, front mic. Kathy.
Hello.
Hey.
Hi. Very nice talk, Bill. And I'm also very interested in the interaction between germline DNA and the somatic cell mutations. And I guess, do you think what goes on initially is a child is treated, and then the somatic cell mutations crop up in just a few of the leukemia cells, and then those grow out so that when it returns, if you will, now the mutation's everywhere, or do you feel like it's in different populations of cells?
Yeah, it's a great question, and the answer is both. And we have data. Our collaborator in Project One, Charles Mulligan, as well as Jude Yang, have both published papers on sort of this evolution from diagnosis to relapse.
Yes.
And actually, there's evidence for both things happening. That is, there are subclones, and they may be very minor, less than 1%, at diagnosis that harbor that NT5C2 variant, for example. And over time, it just grows out because it's the one that's resistant.
Right.
There's another subset, maybe almost equal in number, that acquire that variant during the course of treatment, that we can't find any evidence that it exists at diagnosis. Now, the better our whole exome sequencing gets and the deeper the coverage, the more we're able to find these very minor clones. So that the answer to that's probably going to continue to change as our ability to detect small subclones at diagnosis improves. Okay. And what we'd like to do, we haven't quite yet figured out how to do it, is we'd really like to pull out all those subclones and test their sensitivity to drug. Yeah. Right now we're testing the gamish, which represents the dominant clone at diagnosis.
Yeah. Thank you.
Back microphone.
I don't know. I think he's just...
Oh, there's nobody. Sorry, Lauren.
Hi, Dr. Evans. Are you going to be testing for NUD-T15 and adjusting the doses of the thiopurines based on that in these patients? Yes.
But we're going to do it in a clinical trial first. We're going to do it in the new frontline ALL trial at St. Jude. We're going to test under CLIA, and we're going to, as currently planned, and it's through the scientific review, but it's still got to go to IRB, but I think they'll approve it. We're going to adjust in those that had the strongest loss of function variants. The STAR4 variant is sort of an intermediate variant, and it's not so clear what we should do. So we want to err on the side of over-treating versus under-treating, but if we need to, we can adjust based on clinical grounds if they're intolerant. But we will prospectively adjust as currently planned based on NUD-T15 and TPMT. Adjust their starting dose, and then we always titrate that dose after that based on their counts. Yeah. I'm glad you got a ticket to get in. That's good.
Thank you. Thank you.
They weren't letting her in earlier. So that's good. You cut me off and we're done.
And you may have one more here.
Hi, very nice talk, thanks. The promoter methylation changes you saw on Cas-P1,
Yep.
did you do whole methylome analysis, and did you see global aberrant methylation levels, or was that specific for the Cas-P1?
No, we did whole genome-wide in terms of the 450K Illumina array, right? And these were the ones related to steroids, right, sensitivity. We find other methylation sites are involved with other genes for other drugs, which makes sense, right? Different enzymes metabolize them, or different targets for those drugs. We haven't done a comprehensive analysis by which we would declare the extent to which hypomethylation is operative in childhood leukemia, but we do know it's aberrant. We made some comparisons to normal leukocytes in these patients, but that's not really a perfect normal tissue comparator for leukemia cells. So some of that's in the paper, in the supplement. It does look like leukemia cells that are resistant based on caspase-1 overexpression, if you compare them to the normal leukocytes in those same patients, it looks like they've lost methylation, that there was methylation in their normal leukocytes, and in that subset that became resistant, for some reason it was hypomethylated, for whatever mechanism, we don't know. There was one reviewer who wanted to know the answer to that, and we finally convinced them that was beyond the scope of the paper.
Okay, thank you
Thanks.
very much.